We're a little bit late. I know a few of you actually aren't coming today because of various qualifying exams and similar things, COVID shots, that kind of good stuff. Before we get started, again, I'm going to apologize. I don't have your exams. I'm not even sure I'll have your exams for Thursday. We'll see. You'll definitely get them by next Tuesday. That's for sure. But we'll see about Thursday. Does anybody have anything procedurally they'd like to talk about with respect to the class? The schedule, old homework, old anything? We're all more or less settled. Homework 8 kind of made some sense. Hopefully, we'll see in a second. Any questions at all? Then let's get started with homework 8 with Jim for problem 1. Jim's on here, right? I thought I saw him.
Yep. Yeah, I'm here. Let me just find the question in the book
You bet.
because I didn't write down that part. But I can share the answer I have. Well, I have it on here actually. So problem 5.2 in the book, find the coefficient of kinship for half siblings and first cousins. I was a little confused by this, whole exercise. But the best I could come up with was, I think for half siblings, it's similar to full siblings in a way, right? Because
it should be different, right? Shouldn't it?
I was just thinking, going back, it's a half chance of passing on the allele from the parent, and then it's a half chance. Maybe I'm missing 1/2 factor here. Maybe
Sounds like you might be missing a half there.
Yeah. And
So first define what the coefficient of kinship is. What is that?
I honestly don't remember what they're describing it as. To be honest, it's the probability of transfer of it being the same across individuals, kind of like an IBS kind of thing.
Yeah, let's be precise about it because there's so many goddamn we're gonna see a lot of these coefficients going back and forth over the next 2 homeworks, and let's be clear on what all of them mean precisely. So this one, Gillespie always writes as fxy, and it might have a name called the coefficient of kinship, but it is the probability of a randomly picked allele from individual X is identical by descent with a randomly picked individual from individual Y. So the procedure is you've got 2 people, X and Y. You close your eyes, you grab an allele from X. You close your eyes, you grab an allele from Y. fxy is the probability those 2 alleles are identical by descent because X and Y have a common ancestor. So for half sibs, I go in and I pull an allele from half sib X. That allele came from one of X's 2 parents. One of X's 2 parents is unrelated to Y. So with probability of half, that allele came from the parent that was related to Y. So I get a 1/2 there. Now I've pulled an allele from individual Y. Again, with probability of half, that allele came from the parent that is related to X, and with probability of half, it came from the other parent. This would be easier with a blackboard. But so I've pulled an allele from X, with probability of half, it came from the parent that's related to Y, and I pulled an allele from Y, with probability of half, it's from the same parent. But that parent has 2 alleles. I might have pulled the same allele twice. That the parent might have passed the same allele to both X and Y, or could have passed different alleles. It passed the same allele with probability of half. So I end up with a half times a half times a half. Does that make sense? So fxy for half sibs is in fact an 1/8, and you have it exactly right for first cousins. It's a 16th. And again, from first cousins, you could do it by that same argument of, so I pull an allele from one cousin, I pull an allele from another cousin with probability half it came from a parent who were going to be siblings, and then trace it back to grandparents. And you will get a 16th by that sort of logic argument. When Wright was doing these problems in pedigrees, in inbred cattle pedigrees, you sort of find that the easiest way to do them actually is to draw all the non-redundant paths between X and Y. The answer for fxy is it's 1/2 times the number of steps in a non-redundant pathway summed over all non-redundant pathways. So if you look at a pedigree and draw all the different ways an allele could be IBD and just count the number of individuals in those pathways, summed over all the pathways, you'll get the right answer. Or you could do it the Gillespie way and just make this sort of probabilistic argument.
So I think what confuses me about it is I was kind of taught to interpret pedigrees as for every degree of separation, there's 1/2 chance, multiplied. But where does the extra half in this situation come from? It's from
the number of nodes.
IBD?
It's the number of nodes here. It's 1/2. It's so for fxy, that's the probability two alleles are identical. Grabbed, it is the number of nodes. So it's a half sib, a parent, a half sib, 3 nodes, 1/2 to the third. So for full sibs, it's a similar argument. It's one full sib back to mom, back to the other full sib. That's an 1/8. But it also could be one full sib back to dad, back to the kid. That's also an 1/8. So you get a 1/4. Because there are 2 different ways you could get an 1/8 for full sibs. And for first cousins, you'll find there's something like 2 different ways you can get 1/32, which is why you end up with a 16th. And that is for fxy, which is the coefficient of kinship, which is most certainly not the coefficient of relatedness. We will see what that is in a little bit, which is a different thing, but may differ from fxy by a factor of 2. So any kind of thing you were taught about the coefficient of relatedness will turn out to be correct for the coefficient of kinship by a factor of 2 off. So you might have been telling me, you might have been saying, I had an algorithm to work out coefficient of relatedness, and I think you might be right. Other questions? We will see more of these as we go along. You're going to end up with effectively 5 things that are going to matter. The coefficient of relatedness, which is going to give us stuff about population level inbreeding. You will hear a coefficient of relatedness, which is going to equal the coefficient of kinship, maybe off by a factor of 2 or something. You are going to see also 3 separate probabilities, which are sometimes called Cotterman coefficients, but we will simply call R0, R1, and R2. And they are the probability these 2 individuals share 0 alleles IBD, 1 allele IBD, and 2 alleles IBD. And we will find that things like the coefficient of relatedness and the coefficient of kinship are easily found if you knew all the individual R0, R1, and R2 probabilities. And next week, we will end up with spending most of our time thinking about R1 and R2, and sometimes something like R1 over 2 plus R2, which was the coefficient of relatedness. I don't know. Questions? So this is all straightforward so far. We've got Taylor up next with problem 2.
Okay, so problem 2 is problem 5.3 in the book, where suppose all of the inbreeding depression in figure 5.3 is due to 1,000 loci with lethal mutations at mutation selection equilibrium. If the mutation rate to lethals at each of those loci is 10 to the negative fifth, we're asked to find the heterozygous effects, missing an F there, of the lethal mutation H. So we're given a formula in the book where we can represent B, which I think is solved for in that figure, as 1.734. Since we're dealing with 1,000 loci here, it's the summation of little bi from 1 to 1,000. And since and that can be written as 2 pi qi si times 1/2 minus hi. If we assume that, God, I got typos everywhere. If we're assuming the loci are identical and p is approximately 1, that pi will drop out, all these I subscripts will drop out, and we can use the equilibrium q hat equals u over hs and sub that in. And so we get 1.734 equal to 2,000 pqs times 1/2 minus h, sub in u over hs for q. P is approximately, assuming here, I assume is approximately equal to 1. We get this form and we can solve for it as, and we end up getting 0.0057.
Absolutely perfect. At least exactly the same as how I did it too. Does anyone have any questions about this so far? We'll talk about it for a second. So to have this right in your mind, so the picture Gillespie showed originally was real human data, I'm pretty sure. And it was estimating the fitness of children, estimating the sort of survival of children born from humans that had known sort of pedigree relationships, that had f of a quarter, meaning they were sibs, having f of an 8, of 1/8, meaning they were half sibs, f of 1/16, meaning they were first cousins, f of like 1/32 or 1/64, meaning they were second cousins, etc. And if you plot the fitness, or like the log of the fitness, I forget which, you ended up with a kind of linear relationship in f, and the slope was estimable, and the slope as a function of f took this shape b, and in here we're assuming that effectively this is due to selection, due to the standard selection formulas. And if we assume that all of inbreeding depression is happening from alleles of large effect, sort of nearly lethals, that H against them can be really sort of vanishingly small, almost about 1 in 200. So the data in humans is consistent with there being a bunch of lethal mutable alleles in humans, a bunch of lethal mutable loci, maybe 10% of the human genome being lethal mutable. And there being, the vast majority of them being incredibly recessive, with only sort of very, very mild effects against heterozygotes. And that might be the entire explanation for inbreeding depression, at least in humans. Just very, very weak selection and a lot of lethal mutable loci. And that this value of H, you'll see, you know now, is almost exactly the value of H Chuck Langley estimated in flies for lethal mutable loci in flies. Again, very, very small h's on the order of 1 in 200. Now, whether there are really 2,000 genes or 1,000 or 5,000 or 10,000, who knows? So this could easily be off by a factor of 10, sort of one way or the other. Questions about this? But this should be your null model. Why does inbreeding depression exist? It's probably mostly loci that are lethal mutable, recessively lethally mutable or close to it, with very, very minor heterozygous effects. Okay, up next with problem 3 is Kiana.
Alrighty. Yes, I am here. Okay. I wasn't super sure about this, but essentially here I approximated large B, like the question says, using QI is approximately UI over HISI. Additionally, when you make this approximation, you're assuming Q is very small. So P is approximately 1, so PQ is approximately Q. So I made those substitutions, plug them in, and I get this formula for B in terms of H. And so essentially, my interpretation of this was that this is the formula, one of the formulas for S that is in the textbook for viability. So what you can see here is that for a value of Fi or for a constant value of A, this decrease in viability with inbreeding does have a slope of minus B, such that S will decrease as B increases. So the textbook mentions that in real populations, you usually have H less than a half. So for the case of incomplete dominance with H between 0 and a half and a positive mutation rate, you get B that is always positive. Additionally, from looking at this up here, the closer the value of HI is to 0, the larger a value of B is for a given sum overall mutation rates. And the larger B is, the larger the corresponding decrease in viability. And so the way that I interpreted this was, as the fitness becomes closer to a half, for a given value of Fi, the viability will decrease less, if that makes sense. So that's how I interpreted this.
That's it. So the inbreeding depression, as we've seen a few different ways, the existence of inbreeding depression implies H is less than a half. As H gets closer to a half, the effect of inbreeding depression goes away. And it seems like it matters less and less. The closer H gets to 0, the stronger inbreeding depression is. So the more recessive a locus is, the more likely it is to show inbreeding depression, the more it contributes to inbreeding depression. The less recessive it is, the more additive it is, the less it contributes to inbreeding depression. So inbreeding depression is sort of being caused here largely by loci that are really recessive, that the effects you're seeing are from the most recessive of loci, which again, from Greenberg and Crow, we expect to be also the loci with the largest S. So to the extent that inbreeding depression exists, it probably is being caused by some number of recessive lethal alleles, effectively, or nearly recessive. And I'm going to show my screen for a second, because I wrote it in a very slightly different format that does show one thing. So can you guys see my sort of stuff here? So yep. When I rewrite B, we have that sum over n loci. I then sort of just replace that as 2n times averages here. And I didn't multiply through by that 2 so that you could see something. If you don't multiply by the 2, what you end up with is 2n mu times 1 over 2h minus 1. 2n mu is the expected number of new lethal mutations per generation. So what you see is the slope of inbreeding depression looks like the number of new lethal mutations per generation times 1/2h minus 1. Again, as h goes to 0, that thing blows up. At h equals 1/2, that's 0. So your intuition is the slope is in fact equal to the number of new lethal mutations times how recessive they are, 1/h basically.
Can you
That's
remind me
really
what
your
is,
full-on intuition.
sorry, can you remind me what is represented by B exactly? I know it goes into the equation for S, but why are we focusing on B here?
So let's see if I have my, I don't have my copy of, do I have my stupid copy of that fake Gillespie?
I went and dug around in the original paper, and it seems like they describe B as the amount of genetic damage that would only be expressed in a homozygote.
Yeah.
And A is the amount that's expressed in natural randomly mating populations. I just wanted to ask if that is correct.
I think, let me, we should be dumber about this. Da, 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 da. This is, come on. Where the hell is this? What problem was this in Gillespie? 5.4,
4.
okay. I'm in the wrong chapter. That's why I can't find the damn thing. Sorry. There we go. All right. No. I don't know why I'm so bad at this. All right, this is now telling me I'm no longer in the meeting, which seems incredibly unlikely. Why can't I see? Stop share. There we go. Now let's try share something else. So this is less fancy than you think it is. What Morton et al. Have is estimates of viability from outbred individuals, from like second or third cousins, or maybe this is second, let's see. This is parent and offspring, this is full sibs, this is
I don't
half.
think we can see your screen.
Yeah,
Dave,
we're
sorry.
not seeing
Can't
it
see my
yet.
screen, figures. All right, let's try again. Oh, this is great. Stop share. Share. Can you see what looks like an image of Gillespie's book now?
No.
You are sharing. That's, I'll just try sharing my desktop. Can you see my messy desktop with what looks like a PDF on it with a straight line?
No, same. It's just blank.
Oh, good. This is going to be fun. All right, guys. Basic. Can you see this at all? Can you see this PDF?
Yep. Now we can see it.
All right. Now let's try share. How about this one?
Yeah.
All right, now we can see it. Okay. So this is the actual data. On the y-axis, we have something like we have viability, and the points are something like parents unrelated. This is something like full SIBs or full SIBs and parent offspring. This is something like half SIBs / uncle niece. This is something like first cousins. This is something like second cousins. Here on the x-axis, I have F, the inbreeding coefficient, the FXY. The probability 2 randomly picked alleles are, no, they aren't. So this is, if this is 0.12, these are half SIBs. These are not full SIBs. These are half SIBs / grandparent grandchild, I think. Anyway, the plot here is real data. And we've simply fit a curve of y equals e to the minus a minus bf. So we've simply fit this data to that curve. So the data is falling off, not quite exponentially, but it's supposed to be, supposedly. And we have fit that curve. And when we fit that curve, the A term turns out to be whatever that is. And the B term turns out to be 1.7. So this is just an empirical fit to how fast viability is falling off as a function of F, the inbreeding coefficient. So if we assume the reason this is falling off with respect to the inbreeding coefficient is because of the difference in mean fitness between individuals who are outbred and individuals who are inbred, meaning all they have is these two genotypes, we find that the difference in fitness between them looks like 1 minus a it has this form. So this slope, B, looks like the contribution of this term summed over all the loci, which is why we had you find, sort of for values of H for this. So the reason this is a function of F is because inbred individuals differ in fitness, from outbred by a factor of minus B times F. 1 minus A is something like the fitness of outbreds. This is something like the fitness of inbreds, 1 minus BA. So there you go. That's so if this is the way in which viability falls off, that slope is explainable as something like the, sort of the combined effects of in fact, that slope is equal to, when I wrote it, we'll try stop share again. Let's see if I can start share. I just write it as the mutation rate to new recessive lethals times their more or less heterozygous effect, 1 over H, how recessive they are. And if they are at a half, there's no such thing as inbreeding depression. The more recessive they get, the more inbreeding depression looks dramatic. And that should be your intuition. That is the strength of inbreeding depression is proportional to the number of loci that contribute times how recessive they are, 1 over H. Questions about this? All right, this is probably got to be close to all we're going to do on inbreeding depression per se. Okay, up next is Carmen with problem 4. Is Carmen with us? Where should I have to go?
Oh, I'm here.
Yep, great.
Let me find it. Okay, hold on. Oh, sorry, wrong one. Okay. So problem 4 was, it asked you to find this probability of selfing, or yeah, the probability of selfing. So it gives you the genotype frequencies over here. I use those to estimate P and Q, and then I use the heterozygote frequency to find F, and I just plug that into the equation to solve for alpha.
Exactly. So perfect. So here, alpha is the probability an organism selfs rather than outbreeds. Gillespie derives sort of really quickly that if an organism selfs at rate alpha, you expect that that fxy, the coefficient of kinship, i.e. the inbreeding coefficient, ought to be alpha over 2 minus alpha. So if you know your favorite species partially selfs and partially doesn't, and you go and you measure your favorite locus and you test it for Hardy-Weinberg and you measure F, you can actually estimate the frequency with which it selfs just from Hardy-Weinberg. And here with these numbers, we're getting a ratio of 1/3. Does that make sense to everybody? This is a trick. This is a cool little result relevant to anyone who, pretty much any of you who work in plants. If you don't work in plants, well, you know, partial selfing probably isn't a thing for your organism. Might be, but it probably isn't. Any questions? All right, up next with problem 5 is Chris.
All right. Share my screen here. Okay, so yeah, so we know that the inbreeding coefficient is equal to the probability of selfing over 2 minus the probability of selfing. And so essentially just kind of plug that in as a formula and graph it. It being the inbreeding coefficient as a function of the probability of selfing, and this is the graph that.
That's it. Yep. So organisms that are obligate selfers appear to be fully inbred. Organisms that very rarely self will have almost no inbreeding. And if you self some fraction of the time in between those two, you get some value in between. All right, clear to everybody? All right, we now understand how selfing affects Hardy-Weinberg. That's, you know, again, if you work in plants, that's probably something you want to know, because a lot of plants are partial selfers. Okay, Zhangji, you're up with problem, whatever we're on, 6, 7? Problem 6, beginning to do population structure stuff.
Okay, so we have 2 loci, A and B, each of which has 2 alleles, big A, little a, and big B, little b, in 2 populations. And we assume that the frequency of big A and big B are both 1/2, and the frequency of the AB gamete is 1/4. And assuming that in the other population, the frequency of big A and big B are both 1/4, and the frequency of the AB, big B gamete is 1/16. And we're asked to calculate D linkage disequilibrium and R square for each of the populations separately. So to compute D, we just have to take the frequency of the A1, B1 the AB frequency of the AB gamete in population 1 minus the frequency of the A allele and times the frequency of the B allele in population 1, and that equals 0. And similarly for the linkage disequilibrium in the second population, we do the same calculation as well, and that's equal to 0. And then we can compute R squared, but since R squared is proportional to D squared over basically the variance of the A allele and the variance times the variance of the B allele, we get 0 for R squared in the first population and similarly for R squared in the second population. So it doesn't seem like there's any linkage between the the two loci.
It's perfect. There is no linkage disequilibrium within either population. There is no reason to think the A and B locus are anywhere near each other in the genome. They are completely unassociated. A is probably on one chromosome and B is on another chromosome. There's just no LD between them. It's all clear? Nothing to see here. These things are completely unlinked. Up next is Young San with problem 7.
All right, let me share my screen. Okay, so here we have two populations. We have a mixture population where we sample half of the population from population 1, and then, and the other half from population B. So the expected PA, which is frequency of a large A allele, is C1 times the expected value of fraction of A allele in population 1 times C2 times expected fraction of A allele in population 2. So it's going to be 0.05 times 0.5 times 0.5 plus 0.5, which is from C2, times 0.25, which is the frequency of A allele in population 2. So that's going to be 0.375. And for the variance,
Do you have any questions about this so far? Because we're going to keep going. This all makes sense to everybody. It is just the frequency in one population times the fraction from that population plus the frequency in the other times the fraction from that population. Nothing fancy here. Just applying our definitions. Keep applying more definitions.
yeah. So for the variance, I just calculate the expected frequency, expected squared frequency of A alleles minus the expected, the square of expected A allele frequency. So that's going to be C1 times squared expected allele frequency in population 1, and also same for population 2. So that make the variance 0.0468 blah blah blah. And then I did the same thing for loci B, but there is a typo. And then for the FST from the book,
Okay.
it can be, sorry, yeah.
Let me make sure I quite have this. So is that the same number I got? I'm not sure it's the same number I got. That's why I'm a little. I left everything as fractions, I'm afraid. We differ by a factor of 4 somewhere. Where? That's an eighth. That's a 16th times a half, which is 1. That's a 16th times a half, which is 1/32. This number here is probably a bit off. This 0.0625. Now just plugging it in, it looks like you should be getting 1/32 there.
Mm-hmm.
It's like 0.03 or something. Anyway, I wouldn't, I think there's probably a calculator error there somewhere.
Or yeah, maybe I missed 0.05
Yeah.
when I was typing in the,
Yeah, these things happen.
typing this one. Yeah.
Anyway, the logic you've written down is 100% correct. Whether the arithmetic is quite there, I don't care. But this logic is 100% correct. And you would find the variance in PA this way. Okay. And okay. So so what formula have you used for FST? You've used one.
So, so for FST, I use this formula, which seems to be the expected heterozygosity in total or mixed population minus the sum of expected heterozygosity in each population times the fraction of samples from each population divided by the
It's not about
expected heterozygosity in the mixed population.
that is not a bad formula at all for FST. The let me share my screen for a second.
Okay.
That is certainly one of the reasonable formulas for FST, but I'm going to use a slightly different one. It'll give us the same answer, but it may be slightly more interpretable. So can you guys see my screen? Hopefully, please. Yes. Can we see it this time?
Yes.
Good. So I calculated the expected value of the, I of course called it the A1 allele as 3/8. The expected value of the B allele as 3/8. We got the exact same values. When I calculated the variance, I got something like 1/32 there, which made the total variance come out to be 1/64. When I calculate the variance of the B locus, I got the exact same value, 1/64. When I go to calculate FST, I'm going to use a slightly different formula than the one you used. It's perfectly valid. But in fact, I'm going to use a formula that Gillespie doesn't even give. One of the formulas Gillespie gives is that FST equals twice the variance in one of the allele frequencies over this term called 1 minus GT, whatever the hell that was, which turns out to be 1 minus the square of the allele frequencies, which we're just going to rewrite all of this as the variance in the A1 frequency divided by the variance in the A1 frequency, the variance between populations, divided by the variance in A1 in the mixture. So this interpretation, the variance between populations in the frequency of A1 times the frequency of A1 in the mixture times 1 minus the frequency in the mixture, this term can be described as the fraction of the variance of the A1 allele that is caused by variance in the A1 frequency between populations. FST is interpretable as the fraction of the variance induced by population substructure. It is the variance in the allele frequency between populations divided by the average frequency in the mixture times 1 minus that. That is the variance in the mixture. This is the variance between populations. It is the fraction of the variance in the mixture that's due to substructure. And that's how you should be thinking of FST. If this bottom term is the total variance in allele frequency, this top term is the variance in allele frequency in the due to substructure, FST is the fraction of the variance caused by substructure. And that's how you should keep it in mind. That's how you should be thinking about it. And of course, so if you plug in values, I get 1/64 as my variances for both of the allele frequencies. I get the individual frequencies. The average was like 3/8 for both of them. So I get something like 1/15 is my value of FST. questions about this? Did anyone get anything different than 1/15 that they'd like to look at some of their calculations for? So FST at the first locus was 1/15, FST at the second locus was 1/15. And Brooke, do you want to show us what you've done for problem 8?
I do. 1 second. Sorry, I was just finishing writing a note. Okay. Problem 8. Okay. I think this might have, it might have a couple errors just based on carryover from the previous problem, but I think it might be okay. Or it might just be wrong. We'll find out. Okay, so we're calculating the covariance and D and R squared. But I think maybe I misnoted something because I think D and the covariance are, they're probably, I don't think they're the same value, but I think at the time that I was doing this, I was trying to understand how the covariance and D were related.
No. This is the whole point of this problem.
Okay.
So this problem is written in a very, it goes a little fast, if you know what I mean. The last week, you will remember that we thought about the A locus and the B locus as if they were Bernoulli random variables. And we made big A equal to 1, or the A1 allele equal to 1, and the A2 allele equal to 0, and the B1 allele equal to 1, and the B2 allele equal to 0, or big B equal to 1 and little b equal to 0. And we calculated what D was, which was the frequency of A1, B1 gametes minus the frequency of A1 times B1. We found that value of linkage disequilibrium D, and we showed that that value of linkage disequilibrium D was also equal to the covariance between random variables A and B. So when we code the locus as a 1 and a 0, we showed last week that the amount of linkage disequilibrium between these two loci is just equal to the covariance between the Bernoulli random variables, if you call one of them 1 and one of them 0. Here I have asked you to do 2 different problems where you were going to get the same answer both times. And I'll show you my version of it in a second. In one of them, I'm having you calculate D in the mixture population. And in the other, I'm having you calculate the covariance between allele frequencies in the mixture. Not the covariance between the Bernoulli values, but the covariance between the allele frequencies. And let me show you my version of this, because it's very similar to your versions, but it just makes all these things like a little bit more separate. So share screen, homework 8. Can you see my homework 8 again?
Yep.
All right. So the first thing I'm going to do is I'm going to calculate the covariance in allele frequencies in the mixture population. So the covariance between the frequency of A1 and the frequency of B1. By definition, that's the frequency of A1. It's the expectation of the frequency of A1 times B1 minus the average frequency of that should be A1 and B1. Those little ones are missing there. So when I created my mixture, I created a mixture of two populations, each with frequency 1/2. And the frequency of A1 in population 1 is 1/2. The frequency of B1 in population 1 is 1/2. So the fraction of this mixture that came from population 1 is 1/2 times 1/2 times 1/2 times 1/2. The fraction that came from population 2 is 1/2. The frequency of A1 is 1/4 in population 2. The frequency of B1 is 1/4 in population 2. And in the mixture, the frequency of the A1 allele is 3/8. And in the mixture, the frequency of the A1 allele is 3/8. So I got 1/8 plus 1/32 minus 9/64. I got the value 1/64. This is the covariance between allele frequencies in the mixture. Now, I also ask you to find the standard LD measures. So the first thing I need to do is find the frequency of the A1B1 gamete in the mixture. Well, that A1B1 gamete is created by taking a 1/2 and 1/2 mixture from population 1 and population 2. In population 1, the frequency of that gamete is 1/4. In population 2, the frequency of that gamete is 1/16. So the frequency of the A1B1 gamete is 5/32. So the linkage disequilibrium, the apparent linkage disequilibrium in the mixture is equal to the frequency of the gamete minus the frequencies of the individual alleles, or in this case, 5/32 is 10/64. The individual allele frequencies are 9/64, or D is 1/64. So that value is absolutely the same between the two. And here we can calculate R squared. R squared is something like D squared over the individual frequencies. And I got a value of R squared is 1/15 squared. All right. What the hell? So I have two loci. In each in their own populations, there is no linkage disequilibrium between them. If I look at the loci in population 1, there is no linkage disequilibrium. If I look at the loci in population 2, there is no linkage disequilibrium. If I create a mixture of population 1 and population 2, I get apparent linkage disequilibrium. And the amount of apparent linkage disequilibrium I get turns out to be the covariance in the allele frequencies from the mixture sampling. So the fact that the frequency of A1 and B1 differ between the populations is inducing covariance. And the covariance in the frequencies averaged across populations turns out to be the amount of linkage disequilibrium I've induced between them, or at least I appear to have induced. This is sort of wild. It says that if, when I go out in nature and I sample individuals, if those individuals actually come from separate populations, it's going to appear to me there is linkage disequilibrium at any two loci whose allele frequencies differ between the two populations. If the allele frequencies differ between the populations that gave rise to this mixture, I'm going to appear to have linkage disequilibrium between them. Does this make sense? We've gone through a lot of algebra with a lot of numbers that are actually identical, and we've done it so that we can develop this sort of feel that linkage disequilibrium in a mixture looks very much like variances and covariances in allele frequencies within the population that gave rise to the mixture. Up next with problem 9 is Julie.
Yes, can I actually ask a question first
You bet.
about problem 8?
Yes,
What
please.
if the subpopulations are not in Hardy-Weinberg equilibrium? How would that change how we calculate like the covariance between PN and PB?
It would, so if there is a lack of Hardy-Weinberg within populations, and or that there is covariance between different loci, it will increase our measures of linkage disequilibrium in the mixture. In fact, what we'll get is something that looks like this, what we have here, plus the sum of the individual pieces. And yeah, that in fact, when Wright does it, he starts thinking about partitioning the variance and allele frequency. Like if at a single locus, he starts thinking about partitioning the variance and allele frequency into the FST, the amount between the amount of variance due to between population differences, and F within populations, sort of the amount of variance induced by inbreeding. And sometimes we'll even add another layer to it, which is why these things have FST, and I don't want to go there really. But if you make complicated population structures, we can think about partitioning the allele frequency variance into more and more different components, if that makes sense, sort of intuitively.
Yes.
All right.
Thank you. Yes, and for problem 9, we just calculated R square, and then from last assignment, we know that NR squares follows chi-square with 1 degrees of freedom. So if we wanted to have a statistically significant conclusion, it has to be larger than this critical value. And then we calculated that N has to be larger than 864.
Perfect. Absolutely perfect. In fact, you even have the right value of R. You have the same value of R squared I do, 1/15 squared. So, you know, again, I won't guarantee neither of us have an arithmetic mistake, but I think that's probably correct. But yeah, so this is this is getting really near the point, right? So again, we have created these two loci, A and B, to be on different chromosomes. They don't have anything to do with one another physically. And if one of these loci is contributing to disease and you type the other locus, you will see, oh, we haven't got there yet, right? I'm sorry. We're still at typing the disease locus here. So all we've, wait, what have we done here? I'm sorry, I've already forgotten what I asked you here. This is just testing LD, right? Yep. So if you have a large enough sample, you will say that there is significant LD between these loci in the mixture. That at 400 individuals, you would have concluded that these two loci are linked, that they are physically near each other, probably in the genome. They are not. You simply have sampled a mixture, and because of variance in allele frequencies at locus A and B, it appears to you as if these loci are physically near each other when they are not. They have significant LD, but that significant LD is caused by substructure, not by physical linkage. Sense so far? And then,
can I ask a question?
Yep.
Oh, so I guess I just noticed that before when we were, they were talking about how you use data to estimate different quantities. It seemed like the actual number of samples that you use is usually doesn't have to be very large, but it seems like this is the first time that you have to take actually quite large sample to get an estimate of something.
Yeah.
Is that the case?
Yeah.
Okay.
For a lot of things, I end up trying to use somewhat realistic numbers, or at least numbers that realistic in the sense of not so terribly dissimilar from what you will encounter in nature. So FST of point, so for this particular problem, what was FST? 1/15th. So it will turn out that an FST value of 1/15th is kind of consistent. It's not quite consistent with people whose ancestors came from different continents, but it's more consistent with people whose ancestors came from opposite sides of the same continent. So an FST value of 0.15 is consistent, say, with you having a mixture of Swedes and Italians in equal frequency, that kind of thing. And an FST of 0.1 of 1 in 10 is pretty much consistent with you having a mixture, an equal mixture of Europeans and Africans, or Africans and Asians. So FST values in the 0.1 to 0.01 to 0.1 are typical values you'll get from studies of human populations, where you've just grabbed people at random. And we will see that you will be able to therefore detect linkage disequilibrium between unlinked loci when you start getting into samples of size 1,000. Large hundreds to small thousands, maybe even tens of thousands. So that will inform our intuitions for when we need to start being very, very frightened that what we're detecting is not association to disease between alleles and disease, but association between unlinked markers and substructure in crazy ways. Does that make sense?
Yeah.
Yep. So up next is Michelle with sort of the next part of this problem.
So number 10 asks us if A is a disease-causing locus and you type locus B, what might you conclude about the position of the disease locus relative to B? And I didn't do any like math, but we just spent all this time discovering how we could falsely conclude there's linkage. So here you might conclude that the B locus is close to the disease-causing locus.
Yep. That's it. That's all I wanted you to see about this. So if there is true association at A and you type B, or vice versa, because you have induced LD between them, if you have a large enough sample size, you will start seeing what appears to be association at the other locus. And so if FST is 1/15th, and it's that way around the genome, sort of for the whole genome, it's on that order, you might start seeing, well, we'll prove it in a minute, but you could start seeing association to almost every locus in the genome caused by nothing more than variances in allele frequency around the genome. If your sample size is large enough, it's on the order of, hundreds to thousands. We've already seen that, sort of like we might very well need 800 or more, maybe 1,000 chromosomes to even detect the LD. But if you can detect the LD, that's the sort of, the decrement in your power at these unlinked, at these other loci, sort of fall off by that kind of factor. So if you have thousands of people, you could start finding it everywhere. And And now, but there's a little bit of a solution here. Mitchell, do you, what am I asking in problem 11?
Let me see. So you're asking if A and B are really on different chromosomes. If A is disease causing, what fraction of the time would you expect heterozygous parents to transmit the B allele?
It is that stupid. Say it. It's as dumb as you think it is.
Is it a half?
You bet. It is that stupid. So there is a statistical test for association called a TDT test. And what a TDT test does is it asks whether heterozygous parents transmit alleles with fraction a half. That test is invulnerable to substructure. It's just a half, full stop. The only way it differs from a half is if the sites you're tracking either cause disease or are physically linked to sites that cause disease. This sort of population structure induced apparent LD does nothing to actual transmission. So while population structure can mess with your ability to associate, you know, when we sort of do these like chi-squared tests of allele frequency, they do nothing for these sort of, they do nothing to these pedigree-based tests, if you do the pedigree-based tests right. So one possible way of getting around these population structure problems in studies where you try to associate alleles to phenotypes is to measure transmission in pedigrees and test it that way. Does that make sense? And I do this, as you see. I ask the dumbest question possible, and it's just so dumb. You're like, is it really? Yes. But the insight is why stuff like the TDT test exists. It, unlike these tests of association, which can be vulnerable to induced LD by substructure, isn't. And you can. It has TDT type tests have their own issues, but substructure doesn't bug them any at all.
I have a question.
Yeah.
I know that when you, for example, do a GWAS and you look for association between particular allele and disease, often you account for the linkage disequilibrium structure of the genome, for example, like right in that ancestry. And so what I'm wondering is, and this might be a little bit too tangential or conceptual, but I guess why LD blocks and LD structure differs across different ancestries, and I guess if that relates to population subdivision.
Yeah. So I probably have done you a disservice by not really doing these, but the right thing to think is, so let's imagine for a second, we have a new, so let's say for the moment, everyone in the population, well, actually, maybe let's not go there. Let's just simplify it a little bit. So you have your A locus and your B locus. And we saw when we first did the first problems in linkage disequilibrium that every generation recombination breaks down LD between your A locus and your B locus. That in each generation, every recombination event, the frequency of the A1, B1 gamete looks like the frequency it did last generation times the probability there was no recombination plus the probability there was a recombination event times, you know, the long-term expectation, the unlinked expectation. So what recombination does is it breaks down the covariance between A1 and B1. It breaks down LD. So if you have two populations that are evolving separately, the recombination events in one population are not necessarily the same as the other. And the total amount of recombination looks like the time back to the common ancestor of the population. You know, it's a function of that. So if you have some population, they have some common ancestor at some point, the total amount of recombination in the history of this population is a function of the time back to that common ancestor. We get another population, the total amount of recombination is a function of the time back to the ancestor of this other population. If there isn't migration between the populations, the recombination events here are effectively independent of there. So the total amount of recombination proportional to the total time. And if one population, say, has had more total time on average to its common ancestor than the other, the total amount of recombination in that population will be larger, and therefore the amount of LD will be smaller. So we learned when we did those coalescent problems, the time to common ancestor is a function of population size or effective population size. So if you have two populations and one of them has a smaller effective size than the other, the population with the smaller effective size will have a more recent common ancestor, less total recombination in their history, and therefore more LD. Similarly, the population with the larger effective size will have more recombination in their history and less LD. In humans, it turns out that all populations have a smaller effective size than populations including Africans. So the effective population size of Africa is the same or greater than, well, it's greater than any sort of population that doesn't include Africans. And you can think of that a lot of different ways, but the dumbest way to think about that is to remind yourself that 10,000 years ago, most of Europe and most of Northern Asia was under an iceberg. And it's not hard to imagine that very, very few people lived under the ice in Europe or Asia, whereas Africa, which remained much more sort of pleasant place to live, had larger population sizes than anywhere else. As a result, in humans, well, actually almost everything, African population sizes tend to be larger than they are anywhere else. As a result, the total amount of variation, S, tends to be larger, heterozygosity tends to be larger, and the total amount of recombination tends to be larger. So the size so the region over which there is large amounts of linkage to disequilibrium tends to be smaller. And it's all just a population size effect.
Thank you.
Does this make sense?
Yes.
Okay, up next with the proof is Linnea. You wanna take a shot at it?
Sure. I didn't actually do it like generic in values though.
Okay.
Yeah. Also, some of my values are wrong because I mistakenly thought that the frequency of A would be 1932, not what you guys had. So, yeah. Basically, all I did was just wanted to compare R squared and FSTA, FSTB. So I just sort of put them on either sides of inequality to see like would they turn into the same thing. So over here we have r squared, then these are the FST terms. And these are actually, I think they're basically identical because A and B had the same statistics. So you could just think of them as if it was one of them squared. And on the other side, we got this here. Cancel the denominator.
Mm-hmm.
So yeah, then we already calculated like FAB and this one as well.
Mm-hmm.
But then I use this value 19/32, which is wrong. But yeah, they turn into the same thing. So I'm not sure if they were supposed to now or not.
They should have. They are the
yeah.
same thing in this problem. They're exactly
Okay.
this problem. And
Oh, okay.
Individual values are correct. This is totally correct. I think yeah, your 19/32 looks a little slightly off to me, but that's okay. But so here I'll share my screen, and this is just plugging values in, and you got it to be true. So I'm going to be slightly more general with the proof, but the intuition is about the same. Oh, I should be able to share screen. All right, there it is. All right, homework 8. Can you guys see my screen? So I start out defining r squared as d squared over all the allele frequencies. And this, because we showed it above, looks very much like the covariance between the allele frequencies caused by substructure. That's what we got above. And in general, the covariance between two random variables, x and y squared, is less than the variance in x times the variance in y. Why is that? It's because, well, the proof of this is effectively the same proof as that r squared is between 0 and 1. It's a kind of a tedious proof, but it's not very hard to show. So this covariance between allele frequencies is less than the individual variance in allele frequencies within their own populations. And my favorite definition of FST is the variance in allele frequencies between populations divided by the variance in the mixture. That's FSTA, that's FSTB. So R squared in the mixture population looks very much like FST at locus 1 times FST at locus 2. It's in fact less than or equal to, but equality is going to hold in a lot of situations. All right. That's the craziest, deepest result we're going to get here. That if allele frequencies are varying around the genome, every pair of loci will appear to have linkage disequilibrium between them, and the amount they will appear to have will be very close to the fraction of the variance at locus 1 due to substructure times the fraction of variance at locus 2 due to substructure. LD between unlinked loci is getting induced all over the genome, proportional to the variance, proportional to how much of the variance in allele frequency is due to substructure. So the intuitive way to think about this is if you're sampling any kind of mixture, the chi-squared test you get at a particular site is that site's effect plus something about the sites that are in physical linkage with that site contributing, plus all the other sites in the genome that might be contributing downweighted by FST squared, downweighted by the proportion of variance due to structure squared. And that'll turn out to be the reason you can start doing things like accounting for population structure by taking your chi-squared values and I don't know, multiplying them by a constant so that they account for the effects of all the other loci in the genome appearing to contribute here. And this will only be really important for those who end up in the field of asking whether they can associate a genotype with a phenotype. If you're interested in asking that question, never ever forget that the effect you're measuring at a particular site looks like that, plus the effects of any kind of induced LD around the genome due to variances in allele frequencies, because you may have sampled more than one population. And real organisms always have a bit of substructure. There is no organism on the planet that freely mates equally with every other organism around the planet. Maybe some lekking bird does, I don't know, maybe. But the vast, vast, vast majority of the species you're interested in has some degree of population structure. So if you are interested in understanding the relationship between genotype and phenotype, don't forget that what you measure here can look like everything else in the genome decremented by FST squared.
So can I ask a question then?
Yeah.
So is there some kind of, are there some like understood values for that substructure effect that in different organisms? So if you do start testing to see if you really have like a mixed population that truly should be subdivided or should be stratified, like how do you go about making that decision for your study?
So there's a lot of ways to do it. So the first thing to do, the dumbest thing to do is to just go about your study in the dumbest imaginable way, where you don't account for anything at all. You just do your study, pretending that there's going to be no problems, and you're hoping that there are no problems. You just go ahead and naively do your study. And why am I so bad at this, guys? So I would like to. Are you seeing what looks like a PowerPoint presentation? You guys see something that looks like PowerPoint?
Yep.
Okay. So what you do is you do your study very, very naively. Pretend there's going to be no problems. And for in all likelihood, you're not going to test one marker. You're going to test a whole lot of things. You're going to test a whole lot of genes. And what you do is for every single gene, every single marker, you get some p-value, right? You get sort of for this, you get a p-value. For that one, you get a p-value. For the next one, you get a p-value. And you order your p-values from, say, the largest, which are numbers like 1, to the smallest, which are kind of tiny. And I know if those p-values are real, if they are resembling if the model I have, if that p truly represents the probability of seeing my data under the null model, that if I have, say, 1,000 data points, I ought to have one p-value that's like 1/1,000. All right, I have another p-value that's like 2 over 1,000. I have another one that's like 3, and so forth. So if I order my observed p-values, and I expect the most significant one to be like 1 over 1,000, the next one to be like 2 over 1,000, I can ask a question whether, say, if you're seeing my screen, whether, say, the observed p-values ordered from the least significant to the most significant, and then maybe taken on a log 10 plot, if my least significant p-value to my most significant p-value represents what I would expect from a null model. And in here, you sort of see that if orange is no association at any site, it sure looks like my observed p-values are very, very, very similar to what I would have expected under a null model. And therefore, I'm not really terribly concerned globally that there's like a lot of substructure going on here, say. On the other hand, if they had started to diverge very rapidly and kept diverging, I might have very well concluded, there looks like there's something systematically wrong between my statistical, there's something statistically wrong about the test. It is not actually capturing the null model I expect it to. And if I'm doing a chi-squared test, I might be able to say measure the rate at which it's departing from the null model and correct that with some constant factor. That method is called genomic control, and it tends to work very, very well. On the other hand, I could have done what you were sort of hinting at. If I have my data, if I can somehow infer, say from their genotypes, who comes from which population, I might be able to do my tests within populations and then combine stuff later in a kind of meta-analysis. If you're Jonathan Pritchard, that's how you do association studies with a set of programs he called Struct and Strat. On the other hand, if what you're doing is, say, the test statistic looks like a linear model, maybe what I do is I include in my linear model a description of the population structure that I've captured via principal components in the genotype matrix. So if I look at the entire set of genotypes, I might be able to find axes within those genotypes that correspond to what population people came from. And if I can do that, I can then say regress out population from my test statistic. And that's generally how most people actually do these sorts of studies. You end up taking your whole genotype matrix and finding axes that correspond to populations and regress those out of your data. Finally, I could say take the entire genotype matrix, convert it into, say, an estimate of relatedness among all my individuals, and do, say, a linear mixed model. That's sort of the final way I might try to deal with these things. But so there are a lot of different ways. But when you see something that looks like your expected values are very, very, very close to your observed, you're not, say, systematically worried about inflation. And that just happened to be on my desktop because that sort of shit just tends to be there. Other questions? Okay. Well, this concludes our single and two locus population genetics. starting next week, starting next Tuesday, everything becomes highly, highly multi-locus. We start, although fundamentally, all the stuff is really just population genetics, because there are so many loci, we're going to even call the field quantitative genetics. And we're going to be deriving all sorts of results in these quantitative genetics frameworks. In particular, we are going to derive the covariance between relatives in phenotype as a function of various ways of decomposing phenotypic variance. And fundamentally, homework 9 is Fisher 1918. I wouldn't read Fisher 1918 to help you with homework 9. It is unreadable. You can't do it unless you can do homework 9. The paper is literally uninterpretable unless you already know the answer. Won't help you. Kempthorne 1955 genetics on the resemblance between relatives could help you. It is very much the approach we're going to take is Kempthorne 55. But I think you will find it easier to just walk through the problems just the way I do them than to Kempthorne goes much faster. Kempthorne goes really, really fast. But this is what we're doing. We are going to derive the resemblance between relatives. And that's a huge deal. You know, that's matters for everything. Questions? Okay, I would, if I were you, if you have time, start homework 9 before Thursday. And on Thursday, I will field questions and I will give as many hints as I can. And I promise you again for the homework particularly at the beginning, I am not trolling you. We are setting stuff up. Like if the problem seems stupid and simple, good. It should seem stupid and simple, stupid and simple, stupid and simple, stupid and simple. Oh my God, that's annoying, but simple-minded. And eventually you are onto the resemblance between relatives through a series of stupid and simple-minded, but oh my God, there are a lot of terms, problems. Okay, see you Thursday.